Today we're going to talk about Chapter 7, Section 1, which is all about how chromosomes impact the inheritance of certain traits. When Mendel first came up with his basic laws of inheritance, he was very lucky he was studying a simple organism, the pea plant, and the traits that he was looking at were located on different chromosomes. However, that's not the case for all of our genes, and so particularly when we're talking about human genetics, things can get considerably more complex than what Mendel came up with, and that's the beginning of what we're going to be talking about today. The first thing we need to discuss are the two different types of chromosomes that we have. They are in two categories. We have the autosomes, and those are going to be any of our 22 pairs of chromosomes that are not involved in determining the biological sex of the individual. The second type of chromosome is called a sex chromosome, and as you might guess, those are going to be the X and Y chromosomes, which are largely responsible for determining your biological sex. So if we look at this picture called a karyotype here, a karyotype is just a picture of all the human chromosomes. We can see numbers 1 through 22, those represent the 22 pairs of autosomes. And the last pair over here happens to be XY, which would make this a biological male. Now, remember that the reason why we have two copies of each of our chromosomes is because humans are diploid, as well as other animals and a lot of other organisms. So that's why we have two of each of our chromosomes. And that also means we have two of these sex chromosomes. You either have XX, and if you have two X chromosomes, you typically develop biologically as female. And if you have XY, you would develop biologically as male. So let's first talk about the autosomes. When Mendel came up with his ratios, they were only true for what we call autosomal traits. Autosomal traits are traits that are controlled by genes that are located on autosomes, meaning not on the X or Y chromosome. And this was because Mendel studied pea plants. All of the chromosomes in pea plants are autosomes. If you look at this pea plant karyotype that you see here, it's very different from the human karyotype that we saw on the last screen. That's because pea plants only have 7 pairs of chromosomes, and none of them are X or Y. They're just numbered 1 through 7. So all of the genes that Mendel studied were on autosomes within the pea plant. There are many traits that are autosomal in humans. In fact, most of them are autosomal. One example would be hair texture, as you can see here on the right. So basically, any gene that's located on chromosomes 1 through 22 in humans would be considered an autosomal trait. So if you're drawing a Punnett square for how autosomes are going to be inherited, it is very easy. You're just drawing a normal Punnett square. You have already made these Punnett squares. This is your default when you draw a Punnett square, is for drawing autosomal traits. So if we're talking about human genetics, a lot of times when we're talking about these autosomal traits, we are mentioning them in the context of a specific genetic disorder. So when we're talking about these disorders, it's important to introduce this new term. If we have a heterozygote, or somebody who has one dominant and one recessive allele for a particular disorder, they can be a carrier if that disorder is recessive. Well, what does that mean? That simply means for somebody like this genotype right here, they're heterozygous. They have one dominant allele, and not having the disease, in this case, is dominant. Having the disease is recessive. And they do have a recessive allele. They carry that lowercase c, but they themselves are not affected by the disease, and they probably have no idea that they are a carrier at all, not unless they happen to have children who have the disease. That is often somebody's first clue that they're a carrier for a genetic disease. Now, most of these disorders that we will learn about and use as examples are going to be caused by recessive alleles. That doesn't mean a disorder has to be caused by a recessive allele. There are definitely dominant genetic disorders, but the dominant disorders are much less common than the recessive disorders. So now let's talk about sex chromosomes and how those genes differ. If a gene for a trait is located on a sex chromosome, meaning either an X or a Y chromosome, we call that a sex-linked gene. Now, we know that in mammals, the Y chromosome, the genes on that chromosome are pretty much exclusively involved in producing male characteristics. So it's going to have to do with the development of the male sexual organs and really not much else other than that. The X chromosome, however, has a lot of different genes on it, and you might have a gene on an X chromosome that has absolutely nothing to do with determining your sex, but is something that every single living human being needs to survive. That's why everybody, male or female, must have at least one X chromosome to survive. If there is some sort of a mistake in meiosis and there's an egg cell produced that doesn't have an X chromosome in it and it gets fertilized by a Y carrying sperm cell, that embryo will never even develop into an actual infant because everyone needs an X chromosome. So most of our genes that are involved with sex-linked traits are located on the X chromosome. Another word for that is X-linked. If it's on the X chromosome, we would say it's an X-linked trait. If it's on a Y chromosome, which is extremely rare, we would say that that would be a Y-linked trait. So whichever chromosome the gene is found on, that is how we would name it, either X-linked or Y-linked. So now let's talk about how to draw the Punnett square for a sex-linked trait. This is a little bit different from a normal Punnett square that you would draw. Because the chromosome on which the gene is located is important in how this disorder is inherited, we must use X and Y chromosomes in our Punnett square. And you'll see why in just a minute once we draw this Punnett square. Now, most of the time when we're drawing a sex-linked trait, again, they are almost always X-linked. So when we use our capital and lowercase alleles, we're just going to write them as a superscript on the X chromosome to indicate that it's the X chromosome that has that gene. And you can see that in this example down here at the bottom. See this female genotype here, we know it's female because it's XX, and each X has an uppercase H superscript, H being the normal version of the allele, and the lowercase h would be the recessive version of the allele, and if you only have recessive alleles, you would have this disorder, which just so happens to be hemophilia. Hemophilia is a blood clotting disorder. If you have hemophilia, your blood does not clot, and so you can very easily bleed to death if you are injured. And so this was a very serious disease before we had reliable treatments for it. So let's do some practice. We have a question here. A male who does not have hemophilia has children with a female who is a carrier. Remember, a carrier is someone who's heterozygous. What is the likelihood that they will have a child with hemophilia? So let's draw our Punnett square. We draw the boxes as usual, but remember we need to use X and Y. So notice that the father, who's listed across the top, he's XY. He does not have hemophilia, so we have a capital H, a normal allele. Nothing is on the Y chromosome because this gene for hemophilia is not located on the Y chromosome, so it doesn't have any H's on it at all. And we know from the question that we are going to have a female who is a carrier. So you can look over here at our list of genotypes. The carrier is heterozygous. So one of her X's has a capital H, one of her X's has a lowercase h. Now that we have our Punnett square set up correctly, we do fill it in just like we normally would. So here are the 4 potential offspring that could be produced. And you can look at each one of those genotypes in the Punnett square, compare it to the genotypes we have listed here, and you can see which of their children have hemophilia and which ones don't. So in this first box, we have a homozygous dominant female. She would not have hemophilia. We have a heterozygous female. She would be a carrier, but she would not have hemophilia. She does not express this hemophilia gene because she has a dominant allele here. We have a male who is normal. He has a capital H, so that means no hemophilia. And we have one potential offspring who has just one lowercase h. There's no capital H here to cancel it out, so that means that that son would have hemophilia. So you can kind of see from that example that males are going to be more affected than females by sex-linked disorders. And it all comes down to the fact that males are XY. So that means every single allele on the X chromosome gets expressed, whether it's dominant or recessive, because there is not another allele present to kind of cancel it out. However, females have two X chromosomes. So if they inherit one recessive disease allele on one of their X's, it's likely that their other X chromosome is completely normal, and therefore she would not have the disorder. She would be a carrier, but she would not have any symptoms. So if we look at the picture of the X chromosome here, these are just some of the known genes that are on that X chromosome that cause certain genetic disorders. Things like muscular dystrophy, certain types of deafness, Fragile X syndrome, hemophilia from our example, color blindness. Those are all very common examples of sex-linked recessive disorders, and they're all X-linked because they're carried on the X chromosome. On the Y chromosome, we don't have very many things, and as you can see, they're mostly involved in that development of the male sex characteristics. So that's our overview of Chapter 7, Section 1, which is all about sex-linked traits. We're going to continue with 7.2 in a separate video, so stay tuned to hear more about non-Mendelian genetics. As usual, if you have any questions, please make sure that you ask in class.